Presented by Caltech.
It's my honor and pleasure tonight to introduce my colleague in biology, Dr. David Prober. David got his undergraduate degree from the University of Manitoba in Winnipeg. Then he moved across the border and to the University of Washington, where he got his PhD at the University of Washington, the Fred Hutchinson Cancer Research Center. He then moved to New York City, where he started a postdoc at the Skirball Institute of the New York University, where he started working on this project of sleep in the fish that he'll tell us about tonight. And then the lab moved to Harvard, so he completed his postdoc at Harvard University in Cambridge, and then he moved here, and he traversed the ranks, and as this month, he's now been promoted to professor. Sorry, I can't talk right now, but the goldfish is having trouble sleeping. I don't know anything about that, but I might learn so I'll talk to you later. Bye. Sorry. You should turn your cell phones off. Anyway, without further ado, I'll turn the podium over to David Prober, who will, I think, answer a lot of our questions, both about us and our pets.
Thanks, Paul. Okay, so tonight I'm going to tell you about the work that my lab is doing to try and understand how sleep is regulated, and we're doing this using a somewhat unusual animal model using the zebrafish. So sleep is a behavioral state that's conserved throughout evolution. We see sleep in animals that we're familiar with, such as mammals, and but also some animals where you may not normally think of them sleeping, such as birds or fish, or even relatively simple organisms, so invertebrate organisms such as the Drosophila melanogaster, fruit fly, the nematode C. elegans, and even a very simple organism, the jellyfish, exhibits a sleep-like state. And actually, a lot of the work on the nematode is actually done by Paul Sternberg, who just introduced me. Now, this evolutionary conservation suggests that sleep is performing some universal and essential function, but we really don't know what that is yet. Now, the amount of sleep that these different animals exhibit varies tremendously, from as many as about 20 hours a day in the pocket mouse to as few as about 2 hours a day in the giraffe. But the basic behavior is very well conserved. Now, as humans, we devote about a third of our lives to sleep. In fact, we spend more time sleeping, about 8.3 hours a day sleeping, than we do at any other activity, including work. So hopefully sleep is doing something that's very useful and very important, but as I said before, we still don't understand what that is. So there's more reason to think that sleep is important. So first of all, sleep disorders are a major burden on society. It's estimated that about 10% of Americans have a chronic sleep disorder, usually insomnia. Things like driver fatigue are associated with over 100,000 car crashes per year and about 1,500 fatalities. And the estimated cost to society, I've seen numbers ranging from about $10 billion to over $100 billion a year. So clearly this is an important problem. Not only is this important in terms of sleep disorders, but actually sleep is involved in other kinds of disorders. So disrupted sleep is associated with an increased risk of many common disorders such as stroke, diabetes, heart disease, obesity, and several neurological disorders. And disrupted sleep actually has been shown to contribute to the pathology of several neurological disorders. Now, despite the importance of sleep, not only for sleep itself, but for its role in these other disorders, we clearly need new therapies to treat sleep disorders based on an understanding of the biology of sleep. Most current drugs that are used to treat sleep disorders, and this is primarily drugs for insomnia, act as what are known as GABA receptor agonists. And the problem with these drugs is that they have relatively poor specificity for sleep. Basically, they shut off your brain. And so it'd be nice to have more specific drugs that target specific sleep regulators, that these would potentially be more safe and more effective, but this requires a better understanding of how sleep itself is regulated. Okay, so there are some very fundamental questions in the field of sleep research that we're interested in pursuing, and these are very basic questions. This gives you a sense of how little we understand about sleep. So very basic questions, so why do we sleep? And how is sleep regulated? And today I'm really just going to focus on the second question, which is what my lab focuses on. So how is sleep regulated? And the basic ideas or the basic questions that we have are, again, very simple, again, reflecting how little we know about sleep. So first, what genes are regulating sleep, and second, what neurons regulate sleep. Now, the class that I teach at Caltech is the first class that graduate students take when they come to Caltech. And what I always tell them is that when you're starting to address a new biological problem, one of the most important decisions you make is the model system that you choose to study that problem. Now, the vast majority of labs that study sleep work with this model system, the laboratory mouse or rat. And there are some big advantages for studying this model. It's a mammal like us, so it's somewhat related to us. And what's really important is that you can precisely measure patterns of brain activity in these animals to precisely define sleep and wake states, to know when the animal's awake and when they're asleep. And so this has been a very useful model for understanding mechanisms that regulate sleep. But there's some significant limitations to the rodents. And the two main ones are that as a mammal, rodents have very complex brains, and this makes it difficult to understand how neurons regulate complex behaviors such as sleep. And second, rodents are not very amenable to what's known as a screen, particularly genetic screens. A genetic screen is basically a big experiment where you ask what are all the genes or what are all the neurons that are involved in a particular process. And this traditionally has been a very powerful approach to understand mechanisms that underlie biological phenomenon, and this is really hard to do using rodents. So in response to these limitations, over the last 10 or 15 years, several groups, including Paul Sternberg's at Caltech, had developed invertebrate model systems for sleep, such as the fruit fly and the nematode C. elegans. And there are several advantages to using these models. So first, these models are very useful for performing screens. And so labs have used screens to identify genes and neurons that regulate sleep in these systems. But there are some limitations to these systems. So first of all, the brains of these animals have very little molecular or anatomical similarity to mammalian brains. And so at the end of the day, while we may understand in great detail how the brains of these animals regulate sleep in those species, it may not be very informative as to how sleep is regulated in humans. Another limitation is that while many genes have been identified that regulate sleep in these animal models, many of these genes lack clear orthologs or clear versions in vertebrate animals such as ourselves. Now conversely, many of the genes are implicated in mammalian sleep, and there aren't very many, but the genes that have been implicated in most cases lack versions of these genes in these simple model organisms. And so there's still some limitations in all these systems to trying to understand how sleep is regulated. So in response to these limitations, my group and a couple other labs have been using Zebrafish and establishing Zebrafish as a new model system to try and understand how sleep is regulated. Now, when most people think of Zebrafish, they probably think of this. This is not what we work with. So we work with these animals. And so this is what they look like. And so people always ask, so how can you tell the male from the female? So the female fish has a relatively rounded belly compared to the male, which is more streamlined. And the male tends to be somewhat more pink in color compared to the female. Okay, so these are adult animals. And the zebrafish has a useful life cycle. So they start out as a single cell, a very small cell, sitting on top of a big yolk sac. And then they gradually increase in size over development. So they're born inside an eggshell called a chorion. And at this stage, they're referred to as embryos. They hatch from those chorions after about 2 or 3 days. And then they're known as larvae. And at this stage, when they're about 4 or 5 days old, they're about 4 millimeters in size, so about the same size as a fruit fly. And then gradually over time, they grow and get bigger and bigger until they're about 1 inch in size at 6 months. And they're sexually mature at about 3 months of age. And so when I tell people that we work with fish, most people think, or they assume that we work with these guys. But actually, all of our experiments are with these little guys here, these larvae that are 4 or 5 days old. So the zebrafish facility that we have at Caltech is a reasonable size. We have about 4,500 tanks. The tanks are organized into what are known as racks. So each of these racks contains 5 rows or 5 shelves of tanks that each have about 2 liters and hold about 6 fish each. And then 2 rows of tanks that house single fish, 1 or 2 fish in them. And we have about 36 of these racks. So in total, we have on any given day in between about 20,000 and 25,000 fish in our facility. So it's a pretty good size. So what do these fish look like? So I just took a quick video of some fish in their tanks. And so usually when you approach the tanks, they think you're coming to feed them, so they get all excited and they swim around and come to the front of the tank. So this is what they look like. Now, all of our experiments are performed, as I mentioned, using young embryos and larvae. And so it's important that we can obtain a lot of these embryos and larvae. And that's actually one of the great benefits of zebrafish is they're very fecund. You can get many embryos and larvae from pair matings. And so what we do when we want to mate these zebrafish is the night before we want to collect the embryos, we put a male and a female together in a tank, but we separate them with a divider. And when the lights turn on in the morning, that's their cue to mate. So they like to mate at dawn. And so we remove the divider. And what happens is that they swim very quickly next to each other. And the male swims right behind the female. The female releases eggs, the male releases sperm, and the eggs get fertilized. So it's very PG-13. And so an important aspect to collecting the embryos is that there's this insert in the tank that has these holes that the eggs fall through, because otherwise the fish would actually eat the eggs. So we have to do that. So I'll just show you quickly what this looks like. And so when they swim quickly together, you can see these little white balls coming out. So those are the eggs that the female's releasing. So they'll do it again. A second. So there's one over there. I think there's one more. There we go. There's a bunch of eggs released there. And if you actually look down the bottom here, you can see that all these little white spheres, these are all eggs that have been released. And so it's actually amazing to me that, even though this doesn't seem like a very precise process, if the fish are healthy, generally 90–95% of the eggs that they lay will actually be fertilized. And as I said, we can get lots of embryos from a single mating. So typically a healthy male-female pair will lay on the order of 400, 500 embryos in a morning over the span of maybe 30 to 60 minutes. And so if we set up a half dozen or a dozen paired matings, we can easily collect thousands of embryos in any given morning. So we can collect lots of animals with these from the fish. Okay, so my lab has been using zebrafish as a model system to study sleep, and we're using them because the zebrafish is a vertebrate like us, and it combines some of the advantages of vertebrates, but also some of the advantages of the simpler invertebrate models, such as fruit flies and worms. And so I'll just briefly go over what some of those are. So first of all, zebrafish are vertebrate like us, so they do have a vertebrate brain that is similar to ours. So zebrafish don't have a complex cerebral cortex like mammals do, but the parts of the brain that are thought to be particularly important for regulating sleep, like the hypothalamus and the brainstem, are well conserved. And in particular, neuromodulators such as noradrenaline, serotonin, histamine, and dopamine, and some neuropeptides like hypocretin that are present in the hypothalamus and brainstem of human brains are also present in similar regions in the zebrafish brain, and these are very well conserved. So this is a good thing. Second, zebrafish, as I showed you in the last movie, they develop externally from the mother, and development is very rapid. And so they actually have almost a complete body plan after only 24 hours of development. So I'll show you a quick movie of what this looks like. So this movie is going to start at the 2-cell stage. So here are the 2 cells that are sitting on top of this ball of yolk. And this movie starts at about 20 minutes after the egg has been fertilized. And so it's going to quickly divide to 4 cells, 8, 16, 32. And then after about an hour or two, there'll be about 1,000 cells sitting on top of this ball of yolk. And they then begin a process known as epibole, where these cells move down to basically ensheath the ball of yolk. So they cover over the ball of yolk. And then they begin a process known as convergent extension, where cells converge on the dorsal side of the embryo, and they also extend in the anterior direction and the posterior direction. And now you can see the development of the eye. Here's the tail, and then here are the somites, so the vertebrae in the trunk. And so after about 24 hours, you basically see what looks like a full vertebrate body plan. So you can actually do experiments on these animals when they're still very young. Now, as I mentioned, we do our experiments when these animals are about 4 or 5 days old. So I recorded a short movie just to show you what this looks like. So these are some 4-day-old zebrafish larvae in a dish. And just to give you a sense of scale, this is a pen. So these guys are very small. And even though they're only 4 days old, they already exhibit complex behaviors. They exhibit hunting behaviors. They can elude capture from animals that are hunting them, and they have all sorts of complex behaviors, including sleep and wake states. So after only 4 days of development, we can study the behavior of these animals. Another big advantage, and this is a large reason why many labs work with zebrafish, is that at the embryonic and larval stages, they're optically transparent. So this is a picture of a living 5-day-old zebrafish larva where we've labeled about a dozen neurons in the hypothalamus with green fluorescent protein. So we can actually make movies and watch these neurons as they develop and manipulate the neurons. It's actually turns out to be really useful for studying how neurons regulate behavior. Another big advantage is that the larvae are very small. So they're about the same size as a fruit fly, about 4 millimeters in length. And so we can actually study behavior in 96-well plates. And so this is an example of one of these plates. And each of these little smudges here is a zebrafish larva. And just for scale, so this is a penny. So in a footprint about this big, we can study 96 animals. And by comparison, in this footprint, you couldn't even study the behavior of one mouse. So we have a lot more statistical power than the typical mouse sleep lab does. And so what we do is we take this plate, and we put it in a box where we turn white lights on during the day and off at night. And then we have infrared lights and an infrared camera that records the behavior of these animals for several days. And what we see is that they're generally active during the day and generally inactive at night. So just to show you what this looks like, so this is a movie of the typical behavior. So these are the larvae swimming around in the wells. You see they have plenty of room to swim around. And these red flashes are where the computer system is detecting and recording their movement. So we simply just record their movement over time for several days and nights. And this is an example of what the data looks like that we get out of this assay. So here we're looking at the locomotor activity of a single larva. And this data is acquired during the 5th night of development and the 6th day and night of development and the 7th day and night of development, so early during development. And what you see is that this individual is relatively active during the day and relatively inactive at night. But it's very noisy, right? So there's some times during the day when this fish is very active and sometimes where it's very inactive. And this is sort of a general problem with studying sleep, is that it's very variable among individuals. In fact, if you were to do a similar experiment on humans, you'd see the same thing, that some people are very active during certain parts of the day but inactive during other parts of the day. And so this actually makes it difficult to study sleep because there's so much variability. And this is where you have a big advantage of being able to study many animals at once. Because if we average this data over 80 larvae in a single plate, now the data is much more robust, where on average they're very active throughout the day and very inactive throughout the night. So this gives us great statistical power for our behavioral experiments. So another big advantage for zebrafish, and really one of the motivating factors for setting up the system, is that unlike rodents, zebrafish are amenable to screens. And so we can use this as an approach to identify genes and neurons and drugs that regulate sleep. And we've actually used the zebrafish for this purpose, and I'll be coming back later to talk about a genetic screen that we performed. And finally, and what's turned out to be really useful for what I'm going to tell you about today, zebrafish are diurnal animals like ourselves. And diurnal means that they sleep at night and they're active during the day. And this is in contrast to the nocturnal rodents that most sleep labs work with, which are awake at night and sleep during the day. And so you can imagine if you're trying to understand how the circadian rhythms regulate sleep and how the light-dark cycle regulates sleep, in some ways zebrafish may be a better model system for human sleep than a nocturnal rodent. Okay, so how do we study sleep? So typically when sleep is studied in a mammal, you can measure patterns of brain activity and precisely define when the animal is awake and when they're asleep. Now, very often you can also tell just by looking at the animal when they're asleep. Now, the model system we're working with, we have a problem, because this is what zebrafish larvae look like when they're awake, and this is what they look like when they're asleep. There's no eyelids. So Paul has the same problem with his nematodes. They don't have eyes, and if you work with Drosophila, it's the same problem. So how do you study sleep in these animals? Well, sleep is a behavior, and EEGs were first identified, I think, in the 1940s or 50s, but we've known that animals sleep for a long time before that, right? And so how do we know that an animal is sleeping? Well, we look at their behavior. And so a series of behavioral criteria have been established to define a sleep-like state. So first, during sleep, usually, but not always, but usually animals are behaviorally quiescent, so they don't move. And this is usually regulated by circadian rhythms, although that's not always the case. And second, this quiescence is rapidly reversible, and this distinguishes sleep from coma or paralysis. There's an increased arousal threshold during sleep. So for example, it takes a larger or stronger stimulus to arouse a sleeping animal than an animal that's just sitting quietly. So this is how you can tell if an animal is truly asleep. And finally, there's homeostasis. So if you sleep deprive an animal, there's rebound after deprivation. So if sleep is doing something important, if you deprive an animal of sleep, they'll make up for that lost sleep afterwards. And so what we found is that if zebrafish larvae are inactive for 1 or more minutes, there's a very large increase in their arousal threshold. So based on this, we define 1 or more minutes of inactivity as a sleep-like state in zebrafish larvae. And what we and other labs have shown is that zebrafish have behavioral, anatomical, genetic, and pharmacological correlates to mammalian sleep. So what I mean by this, so for example, we did a screen of drugs. We screened 6,000 drugs. for their effects on the zebrafish behavior, which includes essentially all the drugs that have been shown to affect mammalian sleep. And what we found is that the vast majority of these drugs had the exact same effect on zebrafish as they have on mammals, suggesting that similar mechanisms of regulating sleep in zebrafish and mammals. We've also looked at genes and neurons that regulate sleep in mammals, and so far every gene that we've tested does exactly what you would expect in zebrafish based on what it does in a mammal. So these results give us confidence that our findings in zebrafish will actually be relevant to how sleep is regulated in humans. Okay, so I'm going to start off now by talking about a couple models that are used in the sleep field that I think are useful for thinking about the questions that are central to the sleep field and how we go about addressing them. So the first model is known as the two-process model of sleep regulation. And it proposes that there are two processes that regulate sleep. So first there's a homeostatic process known as process S. This process increases with time spent awake and decreases with time spent sleeping. And you can think of this as responding to internal cues for sleep need, and basically your increased sleepiness as you're awake. Now there's a second process known as the circadian process, or process C, and this process responds to environmental cues, basically tracking the Earth's 24-hour day-night cycle. And you see this oscillation over the course of 24 hours. And what the circadian process does is it ensures that an animal sleeps during the appropriate time. And so sleep occurs when the homeostatic process crosses some critical threshold and if you're in the correct phase of the circadian cycle. So for diurnal animals such as ourselves and zebrafish, sleep occurs at night. For nocturnal rodents, that sleep occurs during the day. And probably most people are familiar with how these processes may interact with each other through the phenomenon known as jet lag. Right? So if you're in California and you fly to Europe, you may be awake for a very long time, so your homeostatic pressure may be very high. But often at nighttime in Europe, you'll have a hard time falling asleep because your circadian clock still thinks you're in California, still thinks it's daytime. And so this is a manifestation of how the homeostatic and circadian systems may be out of sync. Okay, so this is one useful model that I'll come back to in a couple minutes. Another model to describe how sleep is regulated is known as the flip-flop model. And this is actually drawn from engineering principles. And the idea here is that you don't want to be half asleep or half awake. You want to be fully asleep or fully awake. You want these to be very stable states. And you want the transitions between sleep and wake to be very fast. And so in engineering, you can actually engineer a switch like this in something that's known as a flip-flop switch. And the idea is you have two populations of neurons. So you have neurons that promote wakefulness and neurons that promote sleep. And what they do is they inhibit each other. Now, if you imagine that you then have these homeostatic and circadian cues, they can impinge upon this balance of wake-promoting and sleep-promoting neurons. So for example, you can imagine a scenario where these homeostatic and circadian cues are acting to either activate these wake-promoting neurons and or inhibit these sleep-promoting neurons. And so if this happens, then these wake-promoting neurons are more active. They're able to more strongly inhibit these sleep-promoting neurons. In turn, because these sleep-promoting neurons are being inhibited, they can no longer inhibit the wake-promoting neurons as well. And at some point, the switch flips, and you have the animal in a wake state, and you're ready to help daddy catch fish in the fish facility. Now, you can have the opposite situation where these homeostatic and circadian cues are activating these sleep-promoting neurons or inhibiting these wake-promoting neurons. The sleep-promoting neurons then can inhibit the wake-promoting neurons, and eventually the switch flips, and you're in the sleep state. In this particular individual, this is a very fragile and rare state that often requires a half hour more driving around in the car, but it does happen. Okay, so we have these models, but the genes and neurons that underlie these models are for the most part still unknown. And so that's what we're interested in studying. So I'm going to tell you two short stories today about work from my lab. First story, looking at the role of melatonin in regulating the circadian control of sleep. And second, I'm going to tell you about a genetic screen that we performed to identify genes that regulate sleep. Okay, so first, melatonin. So this is the model I just showed you. So again, there's a homeostatic process that increases with time spent awake and dissipates during sleep. And there's a circadian process that determines when, during the 24-hour day-night cycle, sleep is permissible. Now, there have been lots of studies in several model systems that have identified a molecule called adenosine as playing a very important role in regulating the homeostatic process. Now, what about the circadian process? So circadian rhythms, circadian comes from Latin, so circa approximately, and diem day. So it's a process that happens with a time scale of approximately 1 day. And the purpose of circadian rhythms is it provides a mechanism to anticipate environmental variations, such as changes in the light-dark cycle or temperature or things like that. And it allows animals to perform behavioral or metabolic functions that are during the appropriate circadian phase. Now, circadian rhythms are widespread throughout the animal kingdom. They've been observed in bacteria, fungi, plants, and animals. And so we actually know a lot about how the circadian clock works. And the foundation for our understanding of circadian rhythms at the molecular level actually originates from work in Caltech, actually from the lab that occupied the same space that my lab now occupies at Caltech. And this was work that was done over 40 years ago in Seymour Benzer's lab, where he had a graduate student named Ron Konopka who decided to do a screen using Drosophila. And so he decided to screen for Drosophila mutants in circadian rhythms. And the way he did this is based on the observations that in Drosophila, eclosion occurs, eclosion is regulated by circadian rhythms. So eclosion is the process where adult flies emerge from their pupal cases. And this occurs preferentially at dawn. So if you quantify the flies that come out of their pupal cases per hour over the course of several days, you see that in normal flies, this is regulated by circadian rhythms, and they emerge with about a 24-hour rhythm. Okay, so what Ron did was he just mutagenized a collection of fish to mutate their DNA, and then looked for flies that didn't eclose with a 24-hour rhythm. And he found 3 mutants. So there was one mutant that he called period 0 that was completely arrhythmic. So there was no rhythm to when they would eclose, it was just random. He identified a second mutant that he called period short that had a 19-hour rhythm. So the rhythm now, instead of being every 24 hours, was now every 19 hours. And he identified a third mutant that he called period long that had a 28-hour rhythm. Now, it actually turned out that these were 3 different mutations in the same gene, which is now known as period, and identifying this period gene formed the basis for everything we now understand about circadian rhythms. And over the last 40 years since this discovery, we now understand how the circadian clock works in very fine detail. So we understand that very well, but we actually know almost nothing about how the circadian clock itself tells the animal when to be awake and when to be asleep. And so we decided to look at this. And so this is work done by a former graduate student in my lab, Avni Gandhi. And so when you think about a factor that may be mediating the circadian regulation of sleep, it should have 3 properties. So first, this factor, the level or the activity of the factor should be regulated by the circadian clock. Second, if you give this factor to an animal, it should put them to sleep. And third, if you take away the factor, you should eliminate circadian control of sleep. Now, it turns out there's a really good candidate factor for this process, and this candidate is melatonin. Okay. And so why is melatonin a good candidate factor for mediating the circadian control of sleep? So first of all, the production of melatonin is regulated by the circadian clock. So if you look in humans, there's low levels of melatonin during the day and high levels at night, and there's a nice circadian oscillation to its regulation. Okay, second, if you give melatonin to people, and if you do this at the right time of day and with the right dose, it does induce sleep. So in this example, people measured the latency to sleep onset during the day, so how long it took someone to fall asleep, to have a nap. And when they gave people melatonin, either 0.3 or 1 milligram of melatonin, they would fall asleep about twice as fast. So you can put people to sleep with melatonin. And on the basis of this data and the fact that melatonin is considered to be a natural product and is not regulated by the FDA, there's been this proliferation of products that you can buy that have melatonin. Things ranging from pills to patches. There's melatonin water, there's melatonin cream. There are sheets that will dissolve on your tongue, and my favorite, these Lazy Cakes relaxation brownies. These are actually taken off the market because kids were eating these as treats and it was putting them to sleep. And so if you go on the internet, if you read the New York Times, or even if you talk to scientists and you ask them about melatonin, they'll say, oh, sure, melatonin is how the circadian clock regulates sleep. And this is sort of conventional wisdom. But if you talk to people who actually study sleep or circadian rhythms, you get actually the conventional wisdom is the exact opposite. They tell you that melatonin has nothing to do with sleep and nothing to do with circadian rhythms. So why is this? So this goes back again to what I said at the beginning of the talk about the selection of the correct model system to study your question. And so there are 2 problems with melatonin and its role in the circadian control of sleep. So first, its production is regulated by the circadian clock, but it's present at high levels at night in both nocturnal and diurnal animals. So if you look in a diurnal animal such as human or in a nocturnal animal such as rodents, there's high levels at night and low levels during the day. So in nocturnal animals, there's high levels of melatonin at night when these animals are awake. And in fact, if you give melatonin to these animals, it doesn't put them to sleep, suggesting that melatonin does not induce sleep in these nocturnal animals. So that's a problem. Second problem is that most of the laboratory mouse strains that people use for their behavioral experiments actually don't make melatonin. These different strains actually have mutations in the genes required to synthesize melatonin, and it's thought that as part of their evolution to adapt to live in the lab, they've actually lost the ability to make melatonin. That's probably related to the role of melatonin in seasonal reproduction, because in a lab setting, you want your mice to be able to mate on a regular basis and not only a couple times a year. So as a result, these mice don't make melatonin, yet they still have circadian control of sleep, suggesting that melatonin is not important for the circadian control of sleep. And there's an even bigger technical problem. If you want to ask whether or not melatonin is required for sleep, what you want to do is take away melatonin. Basically, you want to compare animals that don't make melatonin to animals that do and see if there's a difference. But if all of the animals don't make melatonin, there's nobody to compare them to, so you can't even do the experiment. So based on this data, the mouse field has concluded that melatonin is not involved in the circadian control of sleep and therefore is not an interesting molecule to study. Okay. So I actually didn't realize when we started these experiments that it's actually very controversial, because the vast majority of labs that study sleep work with rodents, and they've decided that melatonin is not important for sleep. Okay, so we decided to look at this with zebrafish. So zebrafish are diurnal vertebrates like ourselves. The circadian clock regulates melatonin production in zebrafish, and if you give melatonin to zebrafish, it puts them to sleep. Okay, so the rate-limiting step in the synthesis of melatonin is catalyzed by an enzyme called ANAT2. And so this is expressed in the pineal gland, which is the organ that produces, or the gland that produces melatonin. In humans, the pineal gland is present deep within our brains. And in zebrafish, it sits right on top of their head. In fish and birds, it sits on top of the head. And so if we perform what's known as an in situ hybridization, which allows us to visualize cells that are expressing this gene, we see that it's expressed at high levels at night and low levels during the day. So it's regulated by the circadian clock. So what we did is we generated zebrafish that have a null mutation in this gene. And these animals don't synthesize any melatonin, and we can now use these animals to ask what happens if you take away melatonin. Okay, so we're using zebrafish to ask what is the function of endogenous melatonin, the melatonin that these animals normally produce. And so the first question we want to know is, do you need melatonin for sleep at all? And so the way we did this experiment is we took animals that have a mutation in this ANAT2 gene. And so the way we set up the experiments, we use animals that are what are known as heterozygous mutants. And so what this means is that these animals, so zebrafish are known as having a diploid genome, which means that they have 2 copies of all their genes, just like we do. And so the animals that we use for these experiments have 1 normal copy of ANAT2, which is represented by this plus symbol, and they have 1 mutant copy of ANAT2, which is represented by this minus symbol. So if you take a female and a male that both have a 1 normal copy and 1 mutant copy of ANAT2, you mate them to each other, what you get is a population of animals where 25% of them have 2 normal copies of ANAT2, 50% have 1 normal copy and 1 mutant copy, and 25% have 2 mutant copies. So these animals don't make any melatonin, whereas these animals do. So we can now simply compare the behavior of these animals to their siblings that act as controls. And so again, we take these larvae, we put them in this 96-well plate, and we track their behavior for several days. Okay. So when we perform these kinds of experiments, we get 2 types of data. So we get locomotor activity and sleep. And so here in cyan, I'm showing animals that have 1 normal copy and 1 mutant copy of ANAT2, and in blue are the animals that have 2 normal copies of ANAT2. So these are animals that make melatonin. So these are our controls. And what you see is that during the day, they're relatively active, and at night they're relatively inactive. And if you look at sleep, they sleep a lot at night, and they sleep relatively little during the day. Okay, so these are our controls. So now we can ask what happens to their siblings that don't make any melatonin. And what we see is very striking. What we see in red, so the red trace represents animals that have 2 mutant copies of ANAT2, so they don't make any melatonin. What we see is that during the day, they look the same as their siblings, their activity levels are normal, and their sleep levels are normal. But at night, they're much more active, and they sleep about half as much. So we can actually quantify this. And if we look over the course of the 5th day of development or the 6th day of development at their sleep, we see that sleep is normal during the day, but at night, the mutants sleep about half as much as their sibling controls. So what this tells us is that melatonin is very important for sleep at night. In fact, if we take away melatonin, they only sleep about half as much. And this is really surprising because considering that the mouse field has concluded that melatonin is not very important for sleep, And even knowing from people who take melatonin to help them sleep, it's not a very potent sleep aid. So it was actually surprising to see such a huge effect of taking away melatonin on sleep at night. Okay, so is melatonin required for sleep? Yes, specifically at night. Okay, so there are some people in the field who say, okay, maybe melatonin can promote sleep. But all it's really doing is phase shifting the circadian clock, essentially tricking the animals into thinking that it's nighttime, and therefore they go to sleep. So if this is true, if you eliminate circadian rhythms, melatonin shouldn't do anything anymore. And we can actually test this very easily in zebrafish. What we can do is as soon as these embryos are born, as soon as they're fertilized, we put them in constant darkness, and we just keep them in the dark while we raise them and during the experiment. And if we do this and look at their behavior, we see that now there's no circadian rhythm, there's no circadian oscillation to their activity and sleep. In fact, if we look at the molecular level and at the cellular level, these animals have no circadian rhythms. Okay, so we've eliminated the circadian clock, we've eliminated circadian rhythms, and now what we see is that the mutants are just always more active and they always sleep less. So what this suggests is that the sleep-promoting role of melatonin is not an indirect effect of changing the circadian rhythms, but rather it's a direct effect on sleep. So melatonin directly affects sleep, not indirectly via the circadian system. Now finally, the question we really wanted to address is whether melatonin is involved in the circadian control of sleep. And so the data I showed you a minute ago, looking at these ANAT2 mutants, what I showed you is that they sleep less at night, but they still do sleep less at night than they do during the day. So there still is this apparent circadian difference in their sleep amounts in these mutants. However, there's a technical issue here, in that there's a phenomenon what's known as the masking or direct effect of light and dark on behavior. And so in diurnal animals such as ourselves and zebrafish, light is very arousing and sleep is actually sedating. And conversely, if you take a nocturnal animal, dark is arousing and light is sedating. So it's possible that the reason that these mutants are sleeping more at night than they are during the day is because during the day the lights are on and that's waking them up. And so the way you can tease out these differences is to perform an experiment where we entrain animals. We raise them in normal light dark conditions. So we turn the lights on during the day, off at night. We do that for the first 4 days of development. And then we put them in the dark, in constant dark, and then track their behavior. And when you do this, because we've entrained their rhythms for several days in light dark, circadian rhythms persist over time. And so if we look at the control animals and look at their sleep, even though they're in constant dark over these 72 hours of this experiment, you see this nice circadian oscillation of their sleep. But now if we look in the mutants that don't make any melatonin, that circadian oscillation is completely gone. Now this was really surprising for us because usually when there are important biological processes such as sleep, you tend to have several redundant mechanisms that underlie that process. So if you take away one of them, there's no big effect. But what this result suggests is that melatonin is the only factor that's required for the circadian control of sleep. So melatonin is required for circadian control of sleep. And finally, we asked how melatonin promotes sleep. And the way my graduate student Avni looked at this was to cross these ANAT2 mutants with lots of other sleep mutants we had generated or tested with a bunch of different drugs that we knew affect sleep. And most of the data was negative, but there was one interaction that she observed, and this was an interaction with adenosine. Remember, adenosine is what I told you before, is involved in regulating the homeostatic control of sleep. And so in this experiment, here I'm just quantifying the amount of sleep at night. And so in red are the homozygous mutants that don't make any melatonin, and these are their siblings that do. And so as I showed you before, the animals that don't make melatonin sleep less at night. But if you treat these animals with a drug called NECA, and what this drug does is it binds to adenosine receptors and it activates it. So first, if you just look at this light blue bar, so these are the animals that do make melatonin. They have one normal copy and one mutant copy, so they make melatonin. And the drug has no effect on sleep in these animals compared to their controls that are not treated with the drug. This is not surprising because we know at night adenosine signaling is fully on, so these adenosine receptors are already maximally activated, so you wouldn't expect the drug to do anything. But the key result is shown here in this pink bar, where we've taken the mutants that don't make any melatonin and treat them with this drug that activates adenosine signaling. And what we see is that it fully rescues the phenotype, so now they sleep just as much as their siblings that do make melatonin. That suggests that what's wrong with the animals here that don't make melatonin is that what melatonin is normally doing is promoting adenosine signaling. If you don't have any melatonin, there's a lack of adenosine signaling, and that's why they're sleeping less. So if we go back to this original model, adenosine has been shown to mediate the homeostatic regulation of sleep, and we've now shown that melatonin is required for the circadian control of sleep. And if we think about this, another way of thinking about this model is that the homeostatic process acts through adenosine to drive sleep. The circadian process acts through some unknown factor to drive sleep. We've shown that this unknown factor is melatonin, and that it promotes sleep, at least in part, via adenosine signaling. And this model is really appealing because it provides a relatively simple way to link the circadian and homeostatic processes to ensure that animals sleep at the appropriate time. Okay, I'm going to switch gears now and talk about how we've used zebrafish to perform a screen to identify new genes that regulate sleep. And so this is work that I began as a postdoctoral fellow together with a colleague, Jason Reel, and we did this in the lab of Alex Shear. But really, the vast majority of this work was done in my lab at Caltech, and almost everybody in my lab had worked on this, but there were particularly large contributions from a previous grad student, Cindy Chu, and two postdoctoral fellows, Daniel Lee and Champreet Singh. And so the rationale for the screen is that what we did is we took a gene, and we did an experiment where we incorporated this gene into what's known as a plasmid of DNA. And this plasmid is special because it contains what's known as a heat shock inducible promoter. And what this allows us to do is to turn on expression of the gene in response to heat. And what we did is we didn't do this for just a single gene, but rather we did this for over 1,000 genes. We generated a library of over 1,000 different plasmids. And what we did is we took each plasmid and injected it into a zebrafish embryo at the one-cell stage. And we co-injected with what's known as a TOL2 transposase. And what the TOL2 transposase does is it causes the plasmid to incorporate into the genome with high efficiency. So this is supposed to represent a chromosome of DNA that the plasmid is inserting into here. And what we do then is we take these embryos, and we raise them until they're 5 days old. We then put them in the video tracker and monitor their behavior. And what's powerful about this approach is that we can use it to test the behavior of individual animals before and after we turn on expression of the gene. So we can compare behavior in the same animals when they're overexpressing a gene that either puts them to sleep or wakes them up. And so there are many ways of analyzing or summarizing the data from the screen, but this is just one way of doing that. So here, this is a histogram showing the number of genes and the total sleep time. And this is referring to how much the animals sleep after we turn on expression of the gene compared to before. And what we see is a nice normal distribution of sleep times, and genes that are over here are genes that cause the animals to sleep less, and genes over here cause the animals to sleep more. And so the first gene that we've characterized in detail, actually, so that in total we tested about 1,200 genes, and about 12 of them gave a sleep phenotype, either more sleep or less sleep. The first gene that we've described in detail is a gene called Nmu, or Nmu. And so here, the animals that contain this heat shock inducible Nmu transgene are represented in red, and their wild-type siblings that don't contain the transgene are in black. So before heat shock, the red and black lines are overlaid here, but then we give a heat shock to turn on expression of Nmu. And what we see is that these animals are much more active during the day, and also the following night, and actually the following day as well. And if we look at their sleep, we see again before heat shock, they look the same, but after heat shock, they sleep much less. So the night after heat shock, there's almost no sleep. They also sleep less the following day, the following day, and the following night. So when we overexpress Nmu, we increase locomotor activity and we decrease sleep. So Cindy's next question was, how is this happening? And so the first thing we looked at was what parts of the brain are being activated when we turn on expression of this gene. And so just to orient you here, so this is a dorsal view looking down from above on a 5-day-old zebrafish larva. If we switch to a schematic view and then show you here where the brain is, so these are some images of 5-day-old larval zebrafish brains. And they're taken from larvae, so in this case a larva that contains the heat shock Nmu transgene and the wild-type sibling. And so what we did is we gave a heat shock, then fixed the animals an hour later, and then performed what's called an in situ hybridization for a gene that's called CFOS. And what's special about this gene, CFOS, is that when a neuron becomes active, it turns on expression of CFOS. And so each of these purple dots that you see here is a neuron that's expressing CFOS, so a neuron that's been turned on. And you see that in the control brain, where we have not turned on expression of Nmu, there's relatively few of these purple dots, whereas in the animal where we have turned on Nmu expression, there's lots of purple dots. And we are most excited about the small cluster of about 5 or 6 neurons here that's bilaterally symmetrical in the hindbrain. And we are most excited about these cells because it's known that there are cells in this part of the brain that are involved in promoting wakefulness. And so Cindy then went on a hunt to try and identify these neurons and figure out what these neurons actually are. And she tested many genes, and at the end of the day, she actually found one that labels these cells. So this is the same experiment, so overexpress heat shock NMU, and now the CFOS cells that are purple here are shown in magenta, and so you can see many of the same regions of the brain that express CFOS here, you see them here as well. But now Cindy is also co-labeled for a gene called corticotropin-releasing hormone, CRH, shown in green here. And what you can see is that this small cluster of 5 or 6 cells in the hindbrain that express CFOS also express this neuropeptide called CRH. So this suggests that when we overexpress NMU, we activate neurons in the hindbrain that express CRH, and that perhaps CRH signaling is required for NMU to induce wakefulness. And so that was the new hypothesis to test. And so what Cindy did was she overexpressed NMU and then treated the animal with a drug that blocks CRH signaling and asked whether this blocks the effect of overexpressing NMU. So in this experiment, we're overexpressing NMU, looking at locomotor activity before heat shock. Both genotypes look the same. We give a heat shock, and the animals that overexpress NMU are much more active than control. What Cindy then did is that after the heat shock, she added a drug that inhibits the CRH receptor. So that's shown in blue and cyan. So first, let's just compare the animals represented by red and cyan. So these are animals that are overexpressing NMU, either not treated with the drug or treated with this drug that blocks the CRH receptor. And so if you compare the red to the cyan here, you see that when we treat the animals with the drug that blocks the CRH receptor, we completely suppress the ability of NMU to make the animals more active. This suggests that CRH signaling is required for NMU-induced arousal. The other interesting thing we observed in this experiment is if you compare the animals that are not overexpressing NMU, so black and blue, what you see is that the animals represented by blue treated with this drug, you can see that the animals represented by blue here are less active than the wild-type siblings not treated with the drug. What this means is that CRH is normally required for normal levels of daytime locomotor activity. Okay, so based on this data, we developed this simple hypothetical model where NMU acts through its NMU receptor. I didn't show you the data today. There's actually 3 NMU receptors. We figured out that NMU receptor 2 is the relevant one. NMU receptor 2 is actually present on these CRH neurons in the hindbrain that are activated when we overexpress NMU. When these neurons are activated, they presumably release the CRH peptide, which then binds CRH receptor to promote arousal. And so now we're working to test this hypothesis. So we're generating fish that have mutations in CRH and the CRH receptor, and we're also trying to generate fish that allow us to manipulate these CRH-expressing neurons in the hindbrain. Okay, so finally, I'll just go back to some of the models that I described at the beginning of the talk. And so there's this 2-process model of sleep control that we've been able to update. So previously it was known that wakefulness drives this homeostatic process through adenosine to promote sleep, that light-dark cycles regulate the circadian process through some unknown factor to drive sleep. What we found is that this factor is melatonin, and that it promotes sleep at least in part through adenosine signaling. But now what we're working to try and figure out is how these happen. So how is melatonin promoting sleep? How is adenosine driving sleep? And how is melatonin interacting with adenosine signaling? And so I have a student who's pursuing these questions right now. In terms of the flip-flop switch that I introduced before, so we have these arrows, but we don't know what molecules are underlying these arrows. And so we'd like to understand what those are. And so one way to start to answer that question is to identify different pathways, different genes, different neurons that regulate sleep, and then try to see how they fit into this model. And so that's what we've used the genetic screen for. So as I told you, we identified this one pathway based on the genetic screen that promotes wakefulness. I haven't had time to tell you about some other work that we've done today, but we've identified this other gene called hypocretin that acts through the hypocretin receptor to promote wakefulness. There's a population of neurons in the hindbrain called the locus ceruleus, which produces noradrenaline that promotes wakefulness. And we've identified other neuropeptides in the screen that drive wakefulness. We've also found peptides that drive sleep. So another gene that we identified from the screen is called neuropeptide Y. This is widely known for promoting feeding behavior, but we've shown that it actually also drives sleep. We've identified another neuropeptide called QRP that acts through a specific receptor to promote sleep. We've identified other genes from the screen that drive sleep. And so one sort of extreme model you can think of is that maybe each of these wake-promoting pathways and each of these sleep-promoting pathways is acting completely independently and in parallel. And there's a whole bunch of different ways you can promote wakefulness and a whole bunch of different ways you can promote sleep. Now, we know it can't possibly be that extreme because we've already found there are some interactions between these pathways. So we found that the ability of hypocretin to drive wakefulness depends to a large extent, although not entirely, on its ability to activate these locus coeruleus neurons. And more recently, we found that the ability of neuropeptide Y to promote sleep results from its ability to inhibit these neurons. Okay, so they're not completely independent, but there are likely some interactions. Now, another possibility, another extreme possibility, is that all of these different wake-promoting systems are funneling into a common wake-promoting system, and all of these different sleep-promoting pathways converge into a common sleep-promoting system, and then there's some antagonism at some level in the brain. And so we think Zebrafish is going to be a very powerful, very useful model system to try and figure out which of these models is correct. And so over the next 5 to 10 years, these are the sorts of questions that we hope to answer in my lab. Okay, so finally, I'll just thank the people who did the work. So all the graduate students, postdocs, and technicians who are currently and formerly in my lab. So it's been a large and a great group. We made, I think, a lot of progress in a relatively short period of time. I'd like to thank our collaborators. So this work was all started when I was a postdoctoral fellow, together with Jason Reel, who now has his own lab in England. This was in Alex Shear's lab. For the genetic screen, the library of genes that we obtained were from Mark Vidal at Harvard. I've collaborated with Joe Feccio at Cornell University to develop new tools to manipulate zebrafish neurons that we use for behavioral experiments. We collaborated with Tomomi Shimoguri in Japan to identify a factor that's required to specify wake-promoting neurons in zebrafish and also in rodents. And then currently we're collaborating with Viviana Gradnaru and Paul Sternberg at Caltech to study genes and neurons that regulate sleep not only in fish, but also in nematodes and in rodents. And we're also collaborating with Scott Fraser, who used to be at Caltech, and now he's at the USC. And we're collaborating with him to study patterns of brain activity during sleep and wakefulness. And finally, just want to acknowledge our funding sources. We've been funded both by private foundations, by some industry collaborations, and also, for the most part, from the NIH. And so I'll stop there, and I'll be happy to take any questions.